CataractCoach.com, podcast series, episode number 121 with Dr. Neel Pasricha, the future of cornea and ocular surface physiology from this amazing young faculty member at UCSF. Welcome back to our Cataract Coach podcast. And today, I have a very special guest who's going to teach us a lot about the cornea, something that I want to know more about because I wasn't smart enough to do a fellowship in it. And that's Dr. Neel Pasricha from UCSF. Welcome to our podcast.
Thanks so much, Uday. It's such an honor to be here, and I'm a huge fan of yours. So thank you again for inviting me.
Of course. Now, before we start, behind you on the wall is a picture of a heart.
Yeah,
Aren't you an ophthalmologist?
Yeah, I know. It's a heart, but it's actually San Francisco as a map. So it's a little bit hipster.
Gotcha. Now, is the Bay Area your hometown?
Not my hometown, but my wife, her whole family grew up here. And so I kind of grew up all over the place. And so people from the Bay Area will appreciate if you're from here, you kind of have to end up back here. So that was why I had a small box where I could ultimately end up.
Oh, no, I totally get it. My high school is like 5 miles or like 8 kilometers from my clinic.
Yeah, that's amazing.
So
it's nice to know
I'm
the
home as
town
well.
and for the town to know you. Yeah.
So take me through your path in ophthalmology. How did you choose ophthalmology? You did undergrad at Hopkins and did obviously very well. You were in med school at Duke, and then you decided you got into ophthalmology.
So I actually thought I'd be a neurosurgeon. That's what I was really into when I was in college. I majored in neuroscience, and I worked with the pediatric neurosurgery department there. And so at that time, Ben Carson was still a practicing pediatric neurosurgeon at Hopkins, and the doctor I worked with worked right across from him. So I was hugely inspired by him, and I thought for sure that would be the path that I'm on. So when I started at Duke, that was still what I was really interested in up until about the beginning of my second year. And that's when my girlfriend at the time, and now my wife, she said, come to the ophthalmology interest group meeting, and there's free pizza. You should come. So I said, okay, I'll come for the free meal. And at that time, Dr. David Epstein was the chair at Duke. Unfortunately, he passed away, but he gave one of the most inspiring 30-minute lectures on why ophthalmology. And I remember I was so inspired after that I said, okay, I'm going to do this, 2-week ophthalmology elective and just check it out. And I was sold. I was hooked. I never looked back at neurosurgery afterwards.
Now, in Dr. Epstein's lecture, what was it that was so compelling?
Just his genuine passion. And it's not that neurosurgeons were not passionate. It's just that the feedback I was getting from them was very consistently, do this if you can't see yourself happy in anything else. Whereas Dr. David Epstein's was just so overwhelmingly passionate about how you can do medicine, you can do surgery, you can take a patient who can't see anything that morning and that afternoon have a patient who's giving you a hug saying, I can see 20/20. That kind of difference making. And I think in med school at Duke, at least, if you missed 2 lectures, you basically missed the entire ophthalmology curriculum. It was a very small curriculum of the whole body. And so I think it was nice to have that 2-week elective where you could really dive deep into it and see how special it is.
I think that's one of the challenges in U.S. med schools. I think the required ophthalmology exposure literally is a day or 2 of lecture at best. And there are no required rotations for most med schools in ophthalmology. So you could do your whole med school, use your MD degree, and you barely can spell ophthalmology.
Exactly. In fact, you usually can't. Yeah, exactly.
Right. So I think, that makes a lot of sense. I kind of stumbled upon ophthalmology as well. And I think a lot of us have. But once you figure it out, you're like, wow, this is great. So we got to thank your wife then.
Oh, 100%. I mean, she was one of those special people where she knew from high school that she wanted to do ophthalmology and was so gung-ho passionate. And so another funny story about that is once I decided in ophthalmology, at Duke, we have a nice curriculum where our 3rd year is a research year. It's kind of an academic year. You get to choose your adventure for that year. And so a lot of us would choose to do research. And so as soon as I identified ophthalmology and wanted to do it, I reached out to a really famous retina specialist at Duke, Cindy Toth. And I said, hey, I'd love to join your lab and do research with you. She says, I've had a med student interested in my lab since 2 years ago. She's already reached out, and lo and behold, it was my wife. And so I ended up, it was a blessing in disguise. I mean, Cindy Toth is amazing, but she connected me to Anthony Kuo, who at the time was kind of a junior cornea specialist at Duke and was doing a lot of exciting intraoperative OCT research in the cornea space. And so I ended up working with him for a year. And he was just such an incredible mentor. And that's what ultimately made me want to go into cornea, was having that year-long experience with him. So my wife is now a retina surgeon and I'm a cornea surgeon. So it all worked out.
But it's funny, it's just what you're exposed to, right? Sometimes.
Right.
Just,
yeah.
I mean, if they didn't have free pizza, you wouldn't have done ophthalmology.
Absolutely not. Right. Exactly.
And so then you did your residency there at UCSF.
Correct. So then after Duke,
and then your
I went
fellowship
to,
passed over.
exactly. So I went to UCSF for residency and really absolutely loved it there. I mean, the training at UCSF for residency was really special. And one of the interesting things about my year was it was the first year they started offering that integrated internship. And so that was a really nice experience where you got 4 months of ophthalmology in your intern year. And then you had 8 months of surgery. So it's combined with the surgery program. And all 5 of us, it was optional for our class, but all 5 of us chose to do it. And it was the best decision we could make, because you got to know, one, all your colleagues on the surgery side, which was really valuable to get to see that side, including neurosurgery, by the way. So I did end up doing 1 month of neurosurgery, and it confirmed that it was not the right career path for me. But then it was really nice to also get that early ophthalmology exposure and get to know the department better as well.
Yeah, for our international listeners in the U.S., ophthalmology residency used to be, like when I did it, total of 4 years, but you do a 1-year internship separately and then do 3 years or 36 months basically of ophthalmology. And now it's all combined into 4 years. And your first year usually is, I don't know, 8-9 months of general medicine, general surgery, and then let's say 3-4 months of ophthalmology, and then the remaining 36 months. So a total of maybe 39-40 months of ophthalmology.
Right.
The full training year. Now, what's the secret sauce? What's the magic in UCSF for residency? Because for 22 years when I was a UCLA faculty member, I would interview every year for the applicants, the med student applicants for residency. And every year we'd lose some incredible good ones that really wanted to come to our program. And they always ranked UCSF higher. What's the magic secret sauce? Why?
You know, it always comes down to the people. I think that's the best thing. But I do think there's certain things about the curriculum that are really special. And probably the most special is our Thursday curriculum. So the way that we do our Thursdays is every week Thursday, our morning is didactics. And we start with this conference that you would really love. It's called 3M, which stands for morbidity, mortality, and microsurgery. So oftentimes we don't have the morbidity, mortality, but we talk about microsurgery. And it's a conference where for 1 hour, we all gather, and it's discussing complicated cases, interesting cases, with a heavy emphasis on the actual surgical videos and going through them.
Oh, I love that.
And it's amazing because you get to see faculty presenting their complications, fellows, residents, and the whole environment is not to, kind of shame anyone. It's all about just teaching. What can we learn from this? What were some tips and tricks that maybe we can take away from this? And you start that as a intern in first year going to these conferences, and now as faculty on the other side of it and coming to these conferences, you just see how much you've grown and how much you can help others and kind of take your mistakes and learn from them. So I think it's a very unique thing about our curriculum. A lot of places will do, these morbidity and mortality conferences maybe once a quarter or once a year, but to do it every single week for 1 hour is really special. And then we have grand rounds, our case presentations. And then for the residents, it's protected time in the afternoon for lectures. So they get lectures kind of back to back to back until 4 or 5 p.m. And they do that every single week on Thursdays for the whole 3 years.
Who's seeing patients on Thursdays?
The fellows and the faculty. And then sometimes the residents will be excused if they're, for example, at our county hospital or a VA hospital, especially the senior resident, the third-year residents.
So the county hospital, VA hospital, they don't schedule clinic or for residents, at least no clinic or surgery on Thursdays?
Not for the whole morning. Yeah.
Wow.
Yeah.
That is very different, very unusual.
It's very different. Yeah. And I think a lot of programs would do, if they had weekly curriculum, it would often be like a after hours thing or like a Saturday. So it's special to have it during the week.
Right. We would have at UCLA, and me think, if you're a first-year resident, there'd be mandatory lectures Monday nights.
Got it.
And then everyone had grand rounds, was basically Wednesday afternoons. And then
Yeah.
that's where we tried to get out of the county hospitals or whatever by that, early afternoon, early afternoon to make it in time back to the mothership here.
Right. And I think that's kind of the norm is it's really hard. You have to, as an institution, prioritize the training so much that you're willing to kind of cut back on these resident run clinics. And as faculty, you have to all be in on it and say, we're not going to, have trainees for that morning session. We're going to be okay with that. So I think that was it's a really special thing about UCSF.
Yeah, I think so. Yeah. Well, the other challenge we had at UCLA was that it's too much driving.
Yeah.
From the main center and basically in Westwood, you got to drive at least 30 minutes to 45 minutes north to go to the big county hospital where I was, all of UCLA Med Center, where I was there for 20 years. And then the opposite direction, 30 or 45 minutes to go to Harbor UCLA. And then you had 2 VAs to cover also. So it was just like, and the ones who suffer the most were probably the retina fellows.
Yes.
Who would sometimes have to drive 100 or 200 miles in a day from one county hospital to the other hospital. I mean, they're spending hours a day in the car.
No, it's rough. And unfortunately, we have that same problem here, Uday. So when people ask me what's the worst thing about UCSF, my number one thing is the driving and the parking, because it is such a hassle in San Francisco. Thankfully, now a lot of our, the UCSF primary facilities are in one campus. But we still have the VA hospital, which is kind of by Golden Gate Bridge. We have our county hospital, which is in the Mission, and then our main facility in Mission Bay. So there's a lot of driving between those for the trainees, but we try and make it so that it's at least you're kind of based in one site for the day, if possible, or at the very most two, because otherwise it's just too much back and forth if you have to go to multiple sites in a day.
Well, it makes a lot of sense. You obviously loved it there as a resident. Yeah. You went away to Bascom. Did you know you were going to come back as faculty?
That I did. And so I was lucky that during my third year, which was right around, it was kind of the peak of COVID, right? Because I graduated residency 2020. And I had talked to Stephen McLeod, who was the chair at UCSF, as a second year resident, saying, I really want to come back. This is my dream program. And thanks to him, I was able to do my interviews, and I was the last faculty interview before the hiring freeze at UCSF for COVID.
Wow.
And so I was able to just kind of sneak in there. And what was nice is then going away to Bascom, I was apart from my wife for 1 year. She was at Stanford finishing her ophthalmology residency. And just knowing that you're going into this, different place for 1 year kind of made it feel like a cornea boot camp. Like I can just say yes to everything. I can do my very best. I'm all in. I really want to just be there to learn, knowing that the job side of things is already taken care of, and I'm going to be coming back to UCSF. And so that was a huge, huge advantage and definitely a huge blessing. And it also really helped for the clinician scientist side, because you were able to start preparing for these, K applications from the NIH early and start to, make it so that starting day one at UCSF, I already had that protected research time from the K grant, which is really nice.
Wow, that's good. Tell me about your research.
Yeah. So my research it's going to sound like it's all disconnected. So I'm going to try and make it make sense.
Sure.
So there's three main research areas that I have. The first, and what I wrote my K grant on, is all about dry eye disease drug discovery, looking at ion transporters on the ocular surface. So that means ion transporters on the cornea and conjunctiva for transporting ions like sodium, chloride, potassium. And what we try to do is see if we activate or inhibit certain ion transporters, how does that change the tear osmolarity and the tear volume? And so that's what I started with when I was a resident, and that's kind of what I initially came in as faculty working on. And this kind of goes back to my intern year, but my intern year, I got to have all this experience with other surgeons. And as part of that, I got to get really close to some of my general surgery colleagues. And so why that's important is when I came back as junior faculty, I went to one of my co-interns' weddings. And it was one of those weekends where I was super slammed with work, but I was like, it's my friend, I got to make it there. So went to her wedding. It was a great time. And I sat next to the head of the UCSF Breast Cancer Center. And she said, hey, you're an ophthalmologist. Like, do you know any cornea specialist who's interested in the corneal epithelium? I said, that's me. Look, I'm here. And she goes, there's these cancer drugs that we're using on our breast cancer patients, and a lot of them are getting these corneal epithelial problems. Would you be interested in learning more about it and trying to figure out what's going on? So that was about 2.5, 3 years ago when I joined. And so that's become a huge part of my research. That's my second research program, is how do we prevent the corneal toxicity from targeted cancer therapies? And specifically, the ones that I'm interested in are called antibody drug conjugates. Have you heard of these before?
No, tell me.
So it's basically traditional chemotherapy. You just give this toxic medicine, right? And the way it works is it kills cancer cells more than normal cells because the cancer cells are replicating faster. So around 2000 was the first FDA approved antibody drug conjugate, which is a really smart idea where they take the toxic part of the chemotherapy and they link it to an antibody. So now you can, for example, exactly, you can target it. So you can look at, for example, in breast cancer, and if they express this HER2 receptor in their breast cancer, you can now give them a HER2 targeted antibody drug conjugate. And in theory, it should be more targeted, less side effects, and better, better for the patient. But even though it's supposed to have less side effects, one of the interesting things we found is about 50% of this whole class of antibody drug conjugates causes really bad corneal toxicity. And it's thought to be this off-target mechanism of how it's doing that. So that's a big part of what my lab is doing, is we're trying to change the way that we grade the toxicity. So how do we classify, what's a grade 1, 2, 3, 4? How do we screen for it better? Because if they get the toxicity right now, the only way to kind of mitigate it is you have to have them either reduce their ADC dose or delay it, or in the worst case, discontinue it. And then the most exciting is we're working on corneal cell cultures and screening repurposed drugs that might be able to block the toxicity. So patients could, in theory, use like an eye drop or something that might be able to keep them on the therapy longer, or allow them to not have all these side effects from it. And so that's been really exciting, and we're kind of moving those things forward. And it's been a fun project to work on because it's this really small niche, but a very big problem that's only becoming more and more prevalent as these drugs get more approvals on market.
Now, what's the mechanism of action? You're giving the systemic targeted treatment, and how somehow it's affecting the corneal epithelium.
Yeah, it's super interesting because one would think, okay, maybe it's because the same receptors in the cancer are in the corneal cells. But interestingly, some of the worst toxicities come from the drugs where they're targeting something that's not even expressed in the cornea. So it has no expression in the cornea, so it's completely off target. And the way that we think it happens is this process called macropinocytosis, which is just a fancy term for the corneal cell kind of non-specifically engulfs the drug inside it. And it's thought to do that because if you think about the corneal epithelium, it's replicating really fast, right?
Sure.
Every 7 days, it's completely turning over. And so the limbal corneal cells have this process where they just non-specifically take things up from the blood. And we're thinking that that's what's happening for the antibody drug conjugates, is it's just being non-selectively taken into the corneal cells.
Wow, interesting.
Yeah.
And so what drugs are you looking at topically on the cornea to help kind of block that?
Yeah, so we're looking at drugs that could be repurposed to specifically inhibit macropinocytosis. So kind of take down, instead of them drinking all this, fluid non-specifically, to kind of reduce that process. But it's a balance, because if you completely block it, the cells will die. And if you don't block it enough, they're going to still get some toxicity. So we're trying to figure out clever ways that maybe we can suppress it, but not to a degree where it's going to harm the cells.
Wow. And then what's the third project?
So then the third one, this is the one that's a reach. Okay, so my first one was all about ion transporters on the cornea and conjunctiva. And the way that they increase tear volume is ions will move into the tear film, and then through osmosis, water will come through these water channels called aquaporins. That's kind of the main water channel throughout the body, including in the corneas. And then my second project is on the antibody drug conjugate corneal toxicity. My third project is we're actually working on developing an antibody drug conjugate that targets aquaporin for pediatric brain cancer.
Oh, wow. Okay. So back to neurosurgery here. Okay.
Yeah. So it kind of goes back to the neurosurgery, neuroscience, but that one is a kind of a crazy story where there's a company called Linden Light that their whole mission is basically to cure pediatric brain cancer. And they reached out to my former mentor in lab who was working on these aquaporin water channels in different diseases. and they said, hey, we saw that pediatric low-grade gliomas have really high expression levels of aquaporin, and a particular type of aquaporin. And my mentor had actually developed a very high affinity antibody against that specific aquaporin. So they said, would you be interested in working on this antibody drug conjugate project that we think could work? And at that time, I had been talking to my mentor, who's kind of semi-retired, lives in Thailand full time now, about this corneal toxicity program. And so he said, well, actually, I know this person who has the experience with aquaporins and is actually very interested in antibody drug conjugates. And so that's how the connection was made. And that's been the newest research project we're working on. And it's super exciting, but I definitely feel like I'm, pitching outside my league right now, but it's been really fun to kind of work on that.
And the nice part here with these funded research, you have like some protected time in the week then.
Exactly. Yeah. So I'm 50% research, 50% clinical and surgical.
Oh, wow. Half and half.
Yeah.
Wow. You're really spending a lot of time in research.
Yeah. It's been really nice because it started off being more like 70% clinical, 30% research, which I love the clinical side, but I felt like the research side, you're just not able to really move things forward. And so now it feels like things can kind of progress in parallel.
Wow, it's nice you find your fit. Everyone's got to find their fit. I mean, for me, even as a resident, research, not exactly my forte. Yeah. I'd much rather be in the operating room or even the clinic. So for me, it's like, number one, I got to be in the operating room. 2 is clinic, and like a very, very distant third is for me doing any kind of research. It's just not my thing.
And I actually really disliked research when I first started. So when I was in Hopkins, I worked in a basic science Huntington's disease lab. And I was doing like all the, basic science techniques like PCR, Western blots, all that. And the project was reasonably interesting. It was on how do we basically prevent Huntington's disease or learn more about Huntington's disease. So I liked the idea, but the day in, day out was so monotonous, and the lab, no one seemed really happy to be there. It just didn't feel like a great, thing that I'd want to do for the rest of my life. And I was very lucky that summer I decided, you know what, I'm going to go try a different lab out. And so I worked in a more translational lab that looked at neurostimulation for stroke rehabilitation patients. And the environment was just a complete 180. People were so happy to be there. It was so patient facing, so immediately translatable. And I just loved it. I had such a great summer doing that. And that was what really made me say, like, this is something I want to be part of my career, is that more translationally focused research where you can take a problem and actually, work with patients and try to figure out solutions.
Right. I like that translational stuff too. Something more direct. I want to see the effect of the research kind of now.
Exactly. I mean, the publications and grants and all that are great, but they're not going to help your patient. You need to eventually take it to be something like some type of pharmaceutical or device or some innovation. And that's the part I'm really excited about, is trying to make that bridge and develop something that's actually going to be used on people.
Now, do you do any of these research projects with your residents? Like when I was a resident many years ago, I'm exactly 20 years ahead of you. I finished residency in 2000. Research was required for residents. You had to do something. Is that same at UCSF?
Yeah, the research is a really big component for the residency. Like we actually just had a resident research day yesterday. Oh, nice. And it was awesome to hear the projects they're working on. It's hard as a resident, I'll say, to do kind of like the more bench translational research. And so a lot of the research projects were more on the clinical database and, looking at big data, that kind of thing. And I don't think it's because residents are lazy. These are still like very high quality, good research projects. It's just more flexible. Like you can do them from home or from, an office. It's kind of hard when you need to do something where you come into the lab Monday through Friday. So there were only, I think, 2 or 3 projects that were more on this like kind of innovative, hands-on, trying new things. So hopefully I'll get a resident soon who will be interested in doing that, but it's hard. It's not easy.
Well, I mean, just the challenge being a resident now is there's just so much more volume to learn.
Yeah.
I mean, it's really just tremendous. But yeah, the reason I actually did was I actually like data more. I'm into math geek. So even now the stuff, I actually still do some stuff, which is AI related, like IOL calcs, with AI input, or big data sets I enjoy. I just,
I saw your video this morning talking about the like how you can use the AI formula to better predict what to do for the second eye. And I thought that was so neat, because right now, if you don't do that, like for me, I'm not using the AI formula. What do? Do you change your target a little bit based on the IOL master and say, okay, it was a little bit myopic here, so maybe I make it a little bit hyperopic target on this eye. So I think the way that you described makes so much sense. So I'm excited to try that out.
Yeah, in fact, the case that I showed was actually emailed to me. Someone emailed to me,
Okay.
and I did the homework and sent him back what I thought the answer was. And I said, well, let me just not waste this. Let me make a little video out of it.
Yeah.
And so I made a video. And the doc who I did it for reported back the patient. He said it was plano minus a coordinates of axis. So basically spot on.
That's amazing.
It's really neat. We're hopefully in process of getting this license actually to have it far more commercially available.
That would be amazing.
Well, we're moving towards the digital OR. Like you said, when you were a resident, or actually a medical student, you worked with a faculty member who was using the OCT.
Yeah.
You look at the cornea during surgeries.
Yeah.
Which is obviously doesn't need it, but it's all going to be integrated. So it's going to be, you'll have a patient in your clinic, they'll be examined on your devices, whether it's your tomographer, topographer, autorefractor, biometer, OCT, all that together. Then the data can be sent to your digital setup in the operating room. So you don't have to mark the eye. The microscope knows exactly where the axis is going to be for your toric lens. Then you'll finish up the case here. It'll do the calcs on the spot, basically. And so at that point, it'll, hone the results for you, tell you what to do. It can even analyze the first eye and tell you again. And then post-op, you put the patient back on the same, let's say, autorefractor or whatever the device, which then sends the data back to the cloud to keep honing your result. You're right. So you don't have to do any manual entry, and it all runs in the background. Because if we look at the idea of honing an A constant,
right.
That's exceedingly rare. It's really exceedingly rare. I mean, today the common thing is the printout comes out and you look at like some calculator like Barrett or Holiday, you're like, you circle, that's the one I want. Okay, next.
Right, exactly. And some people take it the next step and they'll do maybe like their first 100 cases. They'll really kind of look in the chart review and figure out what happened. But those are like the 1%. And even then, it's only about 100 or so cases that one can actually keep up with that and do it. So it's so nice to be able to have something that's just automatically doing it as you go and getting, instead of hundreds of cases, thousands and tens of thousands of cases.
Right. Yeah, there's a power in big data. And then one of the things we looked at was we have a lot of surgeons who use our IOLcalc.com website and submit data and even give us post-op results. And now when we have an unusual eye, axial length, let's say 33 millimeters, really myopic.
Right.
Well, you may only have a couple in your practice, but when you do the big grouping of all surgeons in our database, well, I got 1,000 eyes that have that kind of axial length. No problem.
Right.
Now we can actually analyze some data.
Yeah.
So
that's going to be really neat. I mean, that's a huge innovation. I'm excited for that.
Well, that's kind of the fun thing in ophthalmology. Everyone just kind of finds their own little segment or pocket of what they really enjoy and just do that.
I know. And there's not one size fits all, right? It's so nice that people can kind of choose their adventure in ophthalmology. And I think that's something that I try to tell the residents and the fellows is that there are so many different opportunities, not just within research, like what you research or don't research, but even just what your Monday through Friday schedule looks like. I think you can really, you're an amazing example of that, right? You're able to do this incredible CataractCoach.com resource and also still be a very busy clinician and surgeon and still do innovation and still work with industry. So there's a lot of different paths that one can take. And you can also change what you do during certain points of your career. Like, I think it's not like you have to do what you start with 20 years from now. You can adapt and change.
Yeah, evolve it for sure. Yeah. I like what you said. Ophthalmology is choose your own adventure. That's brilliant.
It is, and it's so nice, right? Like some people want to do clinic and surgery. That's what they're passionate about. That's what they love doing, and I love that too. I love the clinical and surgical side. But I think if that was my Monday through Friday, I could maybe do that happily for about 5 to 10 years. And then I'd start to feel like, okay, I'm getting either burnt out clinically, or I just want to do something different. I want to kind of have a little bit more, something on the side that I'm working on. So it's nice that I kind of am able to do that now. And this is the first time for this past year where I really feel like that balance has been made, where it feels like it's a good balance for me of clinic, surgery, research. It's been good.
So how's that split up in your week, basically? So you said about half your time is spent on research and then half is clinical surgical. You do have a half day a week in the OR, 2.5 days, how much clinic? What do you do?
So I do Mondays is research, which I like because you can kind of meet with your research team. So I have, 2 postdocs and 2 research assistants. So we get to meet together on Mondays, go through the plan, go through some data. Tuesdays is OR. And that, my team knows that Tuesday Neel is usually not available or, only available at like 5, 6 p.m. whenever the day ends. Wednesday's clinic. So same idea there. I'm kind of missing in action in clinic on Wednesdays. And then Thursday mornings, that didactic portion. Okay. So we have all our, 3M conference, grand rounds, case presentations. And then that afternoon is research. So that's usually when I'm checking in again with the team, going into the lab, kind of following up on things. And then Friday's another research day. And so that's when we kind of wrap up and figure out what we're going to do and, how the week's experiments went.
Oh, it sounds like a nice balance, but obviously it took you a couple years to kind of straighten it out and get it in the direction you wanted to be.
Yeah, and I wish I had listened to some of the advice a little bit more closely when people told me, don't commit to too many things early on. Right. And I think it's a common problem is we get so excited about doing everything, right? You come out of training and you just feel like, I want to do all of it. And so I came out of Bascom Palmer, coming back to UCSF saying, I want to do peds cornea. I want to do county hospital resident teaching. I want to start a research lab. And what I ended up finding is that you can do those things. You can kind of, spend time doing all of it, but you just feel like you're not doing any of them well enough.
Right. There are only so many hours in a week.
Yeah, exactly. And it was before I had my daughter. So that also, once you have a newborn, you really can't do these things.
No, I totally get it. I think when you're a resident or a fellow, when you're young and you're training, I think the good move is to just kind of lean towards saying yes to everything. Don't turn down any opportunity. Say yes, 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 yes. And then when you become attending level or consultant level or professor level, at the beginning, ease up on that a little bit. Do fewer yeses, add in some maybes in there, add in a couple no's. And then as you get more progressed, you got to learn to be able to say no. And in a nice way. I usually, if people ask me, hey, would you help me with this project or the other? And sometimes I'll have to say, sounds like a great idea. What a great project. But at this point, I don't have the bandwidth to give your project the time that it deserves.
Right.
And I don't want to shortchange you.
No, and I think that feels bad maybe the first couple of times you do it, but then you realize it's actually the most respectful thing. And people actually, I think counterintuitively, start to respect your time more when they know that you yourself respect your own time, right? Like if you are the type of person that says, I'm only going to commit to something if I know I can do it really well and give it my energy, they respect that about you. And they won't ask you to, join the QI committee or, do these things that maybe you're not so directly passionate about. And they'll kind of come to you for more of the big asks that you want to do that align with you. And that was something I was definitely not good at doing early on. I was just like, yeah, I'll do it. It sounds great. And it's been harder, but definitely it's made a big difference to be able to kind of say no or say, hey, I need help with this.
Yeah, it's one of those lessons where you try to teach someone this, but sometimes you just got to learn on your own. But yeah, I think now I'm very, very, very selective. Like even for working with companies in ophthalmology, big companies, for most I'll say no.
Yeah.
So I'd rather now, I know that I don't necessarily need to work with a big, huge company, one of the big leaders of the company. I'd rather work with a smaller startup.
Yeah, it's an interesting thing because that's a whole different side of things that I feel like doesn't get really talked about is how do you even interact with industry in ophthalmology? And I wish that was part of our curriculum because as soon as you become faculty, companies will come to you or you'll start to be interested in working with them. And no one really prepares us for that well. And so I feel like at UCSF in residency, it was kind of an interesting time where it was basically like no industry, we were very Alcon focused in the OR and in clinic, which is great, but, no other companies. And then if we were doing like a wet lab or something, no lunches allowed, like nothing, even as a resident. And then in Bascom Palmer, one of the biggest, breaths of fresh air there was you had access to every toy you could think of, and it was not that, like whatever company paid you the most or that kind of thing, but it was just, every single company had their products there, and you could get to choose which ones you wanted to use. And I loved that. I loved being able to have access to it, learn it, see it, and get exposure to it. And so coming back to UCSF, I think the atmosphere here has changed a lot for the positive, where it's really viewed as a partnership. Like, we need industry, they need our input, or they should have our input. And together, it kind of feels like a nice, healthy relationship where you're kind of all in it together. Because ultimately, everyone's trying to achieve the same goal of what can be the best for our patients. And so if you work together with them, that's great. But there's a balance, right? If you don't want to say yes to everything, even when industry asks, because some of them are just not maybe worth your time, or not kind of aligning with your career goals.
But I definitely agree. You got to bring back the free meal, the free pizza, because you never know. Look, you may attract some incredible talent who only came for the free pizza, and next thing you know, that person's doing big things in ophthalmology, right?
I think it was a non-sponsored lunch event, but yeah, whatever gets the pizza in, we need it.
Right. No, I think it's important to have those opportunities, and I think to work with industry is really easy.
Yeah.
Just do what you think is best.
Yeah.
And only talk about products that you actually use, you actually believe in, you actually think are great. That's it. Yeah. Kind of the key take-home message for working in public ophthalmology, or key opinion leader, all that stuff is, it'll take you 10 years to build credibility.
Yeah.
10 minutes to lose it.
Yeah.
Lose it. And that's it. Just be true to yourself. And that's it.
That's true.
From that,
and thankfully, especially at Bascom Palmer, there were a lot of mentors who I felt like did a really good job of exactly what you said, where they worked with industry extensively, but they were so true to themselves, and they would never say something that they didn't agree with. And they would never talk about a device that they haven't used in their own patients first. Right. And I think that's really important. But I think for me, I've realized, like, sometimes you get these, surveys, for example, where it's like, oh, talk to us for an hour about corneal scarring. I love corneal scarring. I'll talk for an hour about it. But then you feel like you're just like answering like yes, no questions, or like giving a number for how many patients you see. And while that might be nice for some people, sometimes I'll finish those and go, why did I do that? Like, I don't need to do, an hour of being questioned like a robot. So it's more fun to have the ones where you kind of feel like you're really adding value to something that's going to help your patients that you're seeing in clinic. So for me, the big thing I've been excited about is working more with industry on how do we solve this antibody drug conjugate corneal toxicity problem, for example. And that's been exciting because you can work with them to figure out maybe better ways to grade and screen for the toxicity in their protocols, and that can help them and help the patients too.
For sure. And going back to your other research, your first project with the dry eye, why is it that the only approved drugs we have for dry eye are just all treating inflammation?
Yeah.
Right.
It's a huge unmet need. I mean, we have in the pharmacies, if you go over the counter, it's all these artificial tears, all these tear supplements. But then you're right, everything on market for the most part is all about the inflammatory pathway. So I think it's a huge unmet need right now on doing something that's going to target that osmolarity component, or target like tear film health component. So there is the nasal spray, the varenicline, the Tyrvaya, that does increase tear secretion through nasal stimulation, but it doesn't really add anything that your tears aren't naturally producing. So I'm excited for some of the products that are hopefully coming out soon that are non-inflammatory targeting, but still improve the tear film. So for example, I have no financial interest, but Alcon has this TRPM8 agonist. So it activates TRPM8 channels. And I'm excited for that because it has two functions. It's going to increase the tear volume. So it's going to increase the tear secretion from the ocular surface, but also it affects the corneal nerves. So it kind of has this cooling, menthol sensation for the eye. And I don't know how it's going to work in patients and how they're going to like it compared to other eye drops, but my feeling is that it's going to do well. It's going to be something that's a nice, option for dry eye patients.
Yeah, I'm always surprised at how big the market is for dry eye. It's like 6-7% of the US population is kind of on the low end estimate. And then these drugs these are $1 billion a year drugs. This is no joke.
Yeah. And we all, to some degree have it, right? If you look at a computer screen enough, or when a long OR day, looking through a microscope, I feel like we all get dry eye. And COVID has been interesting because during the pandemic, so many people ended up really getting that more severe dry eye. So I think it's something that's across the board a big problem. And we still don't have a great solution, right? Like you look at the dry eye charts on like how to classify it, it's confusing. Like, most people just want something where they can say, what's a eye drop or a few options that are going to really work. But right now it is a very complex umbrella term for so many different pathologies. What is dry eye? It's not just one thing. It's really, really an umbrella.
And now even more broad, just ocular surface disease. I mean, it couldn't be any broader than that. The whole surface, everything.
Yeah, it's a big open market right now.
Yeah, I think it will be a lot of progress in in the time to come, in the years to come.
I think so. I mean, if you look at these innovation conferences, my wife and I joke, but it's like, 2/3 of the audience is there for retina. It's like a very small portion is there for like anterior segment. But I think if you look within anterior segment, dry eye and ocular surface diseases are definitely attracting interest. And so I think there will be a lot more innovation to come for that.
Yeah, for sure. I mean, part of the reason why retina is so popular now is honestly, it's the pharma side. And one of the things that's kind of always a little bit saddening to me is when you see that, okay, 90+% of the payment from Medicare is going to the drug company, and like a single digit number is going to the ophthalmologist for doing all the work and taking all the liability.
Right. Seems unbalanced.
Yeah, just a little bit crazy, but luckily I'm not on the business side. I like the surgical side, the clinical side far better.
Exactly. What are you excited about innovation wise for anterior segment?
Well, I invest directly and get involved with the things that I think are huge. Yeah. So one is AI-based things such as the way to do lens calculations. Again, my disclosure is I'm part of Advanced Euclidean Solutions. We have patents in this area, U.S. patents and international. Awesome. Second thing I think is really amazing is what's coming is robotic cataract surgery. And I'm involved there as well. So I have another disclosure there with Horizon Surgical Systems. I think that's a really neat topic. I think at some point we have to get accommodating IOLs.
Yeah.
Again, I'm involved there with another project, but I mean, time will tell. But I think for me, at least, my bias is I like those kind of things. I'm not really big into drug research. It's not my cup of tea. But I like things that just directly impact surgery. So the lens calc stuff with the AI, the accommodating IOLs, robotic cataract surgery, these are kind of my big dream project. Plus, I love teaching. So not only CataractCoach, I don't know, your wife may know, we launched Retina Rounds a couple months ago.
Oh, it's a daily ritual. So she watches Retina Rounds, I watch CataractCoach, and our daughter is sometimes with us, kind of looking at both the screens, soaking it in. So but no, I think it's amazing that you're able to have this, educational resource, because that's exactly how people learn. Like, and especially the millennial generation and younger generation, like when we want to learn a new surgical technique, I'm not going to a textbook to learn what are the tips and tricks. I'm going to CataractCoach or YouTube and trying to figure out what are some videos that can kind of help explain different techniques. So, congratulations to you on building such a useful tool that I think is going to be only more useful with time. And I'm curious to see what AI does with it too, because now that you have this amazing database of thousands of videos, I'm sure AI models can basically learn from it too and figure out, okay, what can we take away, or like the key things for cataract surgery or the key steps for different new procedures. So it's exciting.
Brave new world coming up. Yeah, I don't know yet. We'll find out. But I think there is a generational difference. So I look at someone young like my son, who's early 20s, and I was changing the headlight on one of my cars. And so I'm looking through and reading instructions. He's like, what are you doing? Here, just pulls up a video. Watch this.
Right.
I'm like, oh, that's like, wow, I feel like an old man.
Right.
I have to read about how to do it. He's just like, just watch the video.
Yeah, you need carcoach.com.
You're right.
But no, it's a really great way to do it. And it's something where, for example, any of us who played sports, like I'm a big football fan. And so I played football throughout high school, and it's just part of any team sport, right? You prepare for your game, you do your game. And then afterwards, for us, our games are Fridays. Saturday was film review. You sit down and you review your film footage, you learn from it, you practice based on it. And so it's so interesting that I feel like surgical training has kind of lagged behind that model where you kind of just talk about it and read books about it. And I'm just so happy that now it's becoming more common to just have these video reviews. And even with the residents, I love saying, let's review your surgical video together. Let's look at that video and go through it and kind of point out things that you might have missed. You have to review the game footage.
Always.
Yeah.
Look, I still do it now.
Yeah, exactly.
If I entered the case, I got to go back and watch that tonight.
Yep, I'm the same way. And we have this system where it sends it to the cloud, but sometimes it doesn't work for whatever reason. So yesterday I had to go in my normal clothes, put on the bunny suit, go walk my way to the, exactly. Get the good old USB drive and find the video footage. But no, it's really important. And going back to one thing you said about the robotic surgery. So that was an interesting thing where when I was at Duke working on the intraoperative OCT research, we were looking at the DALK procedure, the deep anterior lamellar keratoplasty. And one of the ways that they do that is they take a needle and you put it about 80 to 90% depth in the cornea and you inject air to separate the big bubble technique. Exactly. So that was my first research project we worked on, is how can we use intraoperative OCT to guide the needle to that 80 to 90% depth and actually get that big bubble result. And I showed it to one of my classmates who's a MD PhD, and he was really into robotics and computer science. And he looked at that and he said, a robot could do this.
Right.
And we tried, generation one was we used a little micro manipulator. So it's something that just kind of moves the needle at really fine distances. And we went into the cornea and the cornea just kind of moved instead of the needle going in. But he's taken that idea, and his name is Mark Dralos, and he's now at University of Michigan, and he's working on these robotic intraoperative OCT guided systems. And I really think that the robotic microsurgery is coming, and it's going to be really neat to see, how much better and how much it can assist surgeons in different procedures.
Right. I mean, today the best you could do as a human surgeon is basically maybe 40-ish microns of precision, which is kind of amazing if you think about it.
That is.
But a robot can do, 2 microns, 3 microns.
Incredible. You can't compete with that.
Right. Yeah. And it's same thing with reaction time, right?
Yeah, exactly.
It's at least an order of magnitude faster reaction time.
Well, I hope I'm still employed.
No, it's going to be like, I don't know if you do refractive surgery at all, but like LASIK is already 90% robotic.
That's true. Yeah. Yeah. No, I don't do any LASIK or PRK. I kind of found out in fellowship that we have to do, as cornea fellows, you have to do a minimum of 10 cases. And most people end up doing more than that. 10 was enough for me, where I realized I really liked it. I mean, it was amazing technology. It was really good results, but I felt more stressed for those 10 cases than I did for the, corneal melts and the K-pros and those types of things, because it was just, I found myself saying, I wear glasses and I'm happy with my vision. And it's not that I don't understand the benefit of not having glasses, but the stakes felt so much higher. And the internal pressure I was putting on myself to kind of deliver the best results for these patients, I just found that like reward versus stress ratio was not aligned for me.
Oh, I love doing LASIK. I did a bunch of LASIK yesterday.
Yeah.
And so the post-ops this morning, and they're so happy. And just it's a beautiful procedure. So cataract surgery is really quite beautiful. I'm a huge fan.
Yeah, I'm a fan, but not of doing it myself. Referring them to my really good colleagues who do it like yourself.
Well, it's like you said, you got to find, your happiness. Choose your own adventure.
No, it's true. You have to, and be honest with yourself about it. Like, I think a lot of times it goes back to the whole being able to say no, right? We want to say yes to everything, and especially when you're a young surgeon, it feels bad to say, oh, I can't do this, or, oh, I need to call someone in for help. But one of the things that really helped me when I was, especially in my first year back from fellowship, was having mentors like Julie Schallhorn, who's one of my colleagues and was my residency mentor. She would just volunteer and say, hey, I can come join you for your OR if you want, if you want me to be there to help out. And I always said yes. I loved it. It was so fun because at the very least, you have a buddy that you can, talk to, and it's like a social thing. But and I found that every case I was learning something from her, and just having her there made me feel so much more confident that, okay, I can do this. I got this. And so now to this day, if I have a really difficult case, I have no shame in sharing the case details with mentors from Bascom Palmer or from UCSF and just saying, how would you guys approach this case? And sometimes even asking like, hey, would you be okay coming in for this case and helping out?
I'll tell you what a small world ophthalmology is.
It is.
So no, I'll tell you. So I met her, Shalhorn, and her sister Erin when they were in grade school.
No way.
Their dad, Steve Shalhorn, was a cornea fellow at USC when I was a med student there.
Oh, wow. That is a small world.
Isn't that just crazy?
Like, I mean, the Shalhorn dynasty is strong. Like, you know, Steve Shalhorn is such an amazing guy. He still comes and does our refractive course for the residents. And yeah, they're just an incredible family. But yeah, you're right. It's a small world. And especially as you get more subspecialized, like the cornea specialists, there are so many connections.
Right. Oh, that's the beauty of ophthalmology. I think we're now you must be mentoring some med students and residents as well.
Yes. Yeah.
What are the challenges facing these new med students, new residents these days? I'm a little bit out of touch with it because I stopped working with residents a couple years ago.
Yeah, I think for the med students, it's, you know, I'm biased because I didn't do med school at UCSF. I was at Duke. And at Duke, one thing I really liked is you had that dedicated 1 year where you could really dive deep into whatever of interest you had, whether it's doing a master's or doing research, something. I think now what's hard is when you look at the med students, at least at UCSF, they don't really have that dedicated whole year unless they're taking additional time off or, you know, doing something on their own time. It's usually these kind of side projects as they're doing their coursework or doing their clinical rotations. And I feel like it's hard to get a really good research experience when you have that kind of limited time for it. And also, I think nowadays it's a lot more common to say, well, if I could choose between something in person versus something virtual, I'm going to choose the virtual option because of the flexibility that it allows. So I think it's a hard thing to get students to be able to do kind of this more innovative research where maybe it's actually more patient facing, more translational, and it's a lot more common to do these chart review type research projects. And I think the same issue is happening with the residents too. It's just really hard when they don't have that dedicated time to be able to do these maybe more impactful, innovative research projects. But I also think what's happening too is there's a lot of pressure on the med students and on the residents to do things like publish, right? Like you have to have publications in order to get to your, for med students to get to your residency, for residents to get to your fellowship. And I don't know how good that actually is for our field, right? Because publications and just looking at like a number of publications is not the best metric of how good a trainee is, right? Like you can publish 12, you know, case reports that don't really add any value, or you could publish maybe one really exciting, innovative, novel technique, but that 1 versus 12, it's a hard thing to compete with the person who's the 12 publications. So I hope we move more towards publications don't matter as much as the innovation and the creativity that the students have.
Yeah, I mean, that's the challenge too, is that if you're a med student, you're first trying to be a med student.
Yeah.
That's your first goal. And if you're a resident, your job is to actually, learn ophthalmology, become a resident.
Yeah.
So this is like research is then on top of that.
Yeah.
And then the poor med students, at least the residents get paid a few dollars an hour.
Right.
The med students are going into debt to pay to be there and to pay to do the research. So it's a little bit crazy. And the other thing, finally, is the relevance of peer-reviewed literature becomes less and less over the years.
Yeah.
Showing a list of here, I've done 500 publications on Medline. That was great for 2 generations ago. The ophthalmologists who are now 80 years old or older or already have passed.
Yeah.
That's just a different thing. Because I've met many ophthalmologists, I won't go into no details here, who are huge publications, terrible in the operating room. Horrendous.
Yeah.
Like I'd rather a senior resident do my surgery than that very esteemed big professor with 500 publications, no question. So there's a balance to all this stuff. And then finally, you want peer review? I'll put up a surgical technique tomorrow.
Yeah.
10,000 ophthalmologists will see it in 24 hours.
Right.
Peer reviewed, buddy.
Right.
Instantly.
Yeah, it's true.
And it's not just here. The other thing too, the problem with peer review too is, and I've faced this with, we had a lot of publications we submitted for our AI lens calc stuff as early as 10 years ago.
Wow.
But the first paper was 2015. The people who are reviewing it are the same old guys.
Yeah.
And it's not cool enough. You got a weird name. You're not part of their club.
Yeah.
No, they're rejected.
Right.
So how's that peer reviewed? They're not my peers.
Right.
Peer reviewed is you put something out online and let the world see it, and they'll review it right here, right now. My video from this morning already got, I don't know, 8,000 views from a few hours.
Wow.
It already has a dozen comments and people giving some feedback. I mean, instant.
It's the truest form of peer review. You're right. And now it's very different. And a lot of times you're right. It kind of stays within the closed circles. Like how many people are looking at, antibody drug conjugates in the cornea? There's not that many of us, right? So it's like your peer group is pretty small when you think about it. I don't know. I mean, I don't know how it's going to change going forward, or if it is going to change going forward. But I think it's important that we try and find out how can we better evaluate people and surgeons and clinicians, because the same thing goes true for when a family member asks you, Uday, like, who should I go to for neurosurgery? We don't go to PubMed and go, who's publishing the most neurosurgery case reports and things like that, because you and I and other doctors, we know that that's not saying how good of a surgeon or good of a clinician they are. What we usually do, or what I usually do, is I go to people at that institute that I know and say, hey, who do you think is the best neurosurgeon? Who do you think is the best cataract surgeon at UCSF? And that's how you kind of get the actual insider details. It's not through Google reviews or PubMed. And so we need to figure out a better way to figure that out and get that information out there.
Well, the other thing too is that sometimes we forget that life is a bell curve distribution.
Yeah.
The same way, if we do the distribution of basic science research, I'm on the bottom of that bell curve. I'm like 2 or 3 standard deviations below the mean. But the problem you get is you get residents, and you've seen this, where there is a bell curve of surgical skill, no question. And you can advance up the curve if you really, really work hard. And some people have more innate or natural talent than others.
Right.
But every year, out of a residency of 8 people, let me tell you, with almost 100% certainty, 1 or 2 will be amazing. Most will be very good. And every year, 1 is a stinker.
Yeah.
And that doesn't matter whether you're talking about a residency here, there, anywhere else. It's every program in the country, even the most esteemed top 10 programs.
Yeah.
Every year, there's just some stinkers there. And then you see in the community, in our surgery center, maybe 40-some surgeons operate there.
Right.
And you can see the distribution of what's the average surgical time. We can even, at the end of the year, we can do an analysis. What % of your cases you break capsular bag in?
Right.
It is a bell curve distribution.
That's true. And I've already been humbled now. I'm only 3 years in the faculty, and I've been humbled by both sides of it, where I think based on paper, oh, this person maybe is not going to be the best surgeon or best clinician or best resident to work with or best fellow to work with. And they're amazing. They really blow me away with how dedicated they are, how hard they work, and how good their skills are. But I've also unfortunately had the opposite, where I think, oh, this person is a superstar. Like, they are amazing. They're going to do great. And then they kind of fall a little short, right? They're just not, they don't have that drive. They don't really feel like they want to be there. And I don't know how to predict who's going to be in that first group versus that second group. It's really hard to tell.
Well, now we keep shooting ourselves in the foot, right? We do crazy things like, when I went to med school, there were grades. There's no more grades in med school. When I went to med school, we got the top students and we got them into AOA, Alpha Omega Alpha Honor Society.
Yeah.
Now certain schools have stopped doing AOA because it's, quote, not fair, even though it's based on merit, some nonsensical reasons. Yeah. So they stopped that. Now there's no more step 1 on the boards. No more boards, just pass fail. So and then in particular, University of California has got some major issues with throwing merit out the window and choosing people based on nonsense criteria instead.
Right.
And so that ends up making it a whole nother challenge. So at the end of the day, I don't know what the answer is either. But we got to actually let the pendulum swing back to the merit side.
Yeah, and I think sometimes it's like good intentions, but the outcome is not maybe a good one. And I think we all need to be better about, if we have an idea that we're like, oh, this could be a really good thing for education, and then we get data that, hey, maybe it's not working or maybe we're not happy. it's okay to be wrong. It's okay to say, like, we tried this, it was a good idea, but it didn't work out. Let's go back to something else and try something different. And I think it's really hard because once things get approved now as like, okay, this is our curriculum, it's really hard to then go back and change it. It's kind of like, no, just keep trying this curriculum that maybe is not working well, and let's see if it gets better.
Yeah, no, for sure. When I was interviewing residents for medical students for residency, I only had like 2 things I was looking for, and I only had 2 questions.
Yeah.
2 things I'm looking for: I want to see enthusiasm, and a real drive.
Yes. Yeah.
And then on the questions, I only had 2 questions. One, I'd say, what have you taught yourself outside of medicine in the last few years?
It's a great question.
Because you got to be able to teach yourself, right? I mean, I taught myself how to do DMEC. And if 20 years ago, it didn't exist.
Right.
And you will do some corneal procedure in the future, 5 or 8 or 10 years from now, that doesn't exist now. You have to teach yourself.
I've already had that experience. I mean, the artificial iris is a great example of that. I had my first artificial iris a couple of months ago. I never learned that in residency or fellowship. It was totally new. And it was just awesome to, one, have the videos, of course, to learn from. But then also, we're such a small community that you can just call people who are premier surgeons in it. So Mike Snyder was just amazing. I sent him an email with some questions. He replied back in great detail and then said, this is my cell phone. Give me a call. We can talk about your case. We chatted for 30 minutes about my upcoming case. And it was just an awesome feeling to be like, we are such a close-knit group that you can do that. And so I think you're absolutely right. You have to be able to learn on the fly. I mean, otherwise you're going to be stuck in your ways and you're going to fall behind really fast.
Oh, for sure. Yeah, we had Mike Snyder on the podcast, and we took a whole hour learning all his surgical pearls, and he gave a couple good videos for us. So brilliant guy. My second question is really easy. Describe to me what would be your perfect day of residency. And if you're smart, you'll describe this program because this is all we got.
Right.
If you describe some other program, we don't have that. We don't have the one where you come in the morning at 8 o'clock and go to order your chai latte, and then no, we got to work. It's county hospital.
Yeah, that's
so your perfect day better be our program.
But see, you can get someone maybe who knows what the day looks like and just kind of preemptively says that.
Well, just saying. But you know what kind of there are programs that are resident programs that will spoon feed you.
Yeah.
And there are others where you're like, no, you got to ask for help and you got to get to work and you got to crank.
Right.
I mean, like, I'm used to doing county hospital system. There's a line of patients at the door every day.
Wow.
There'll be 10 or 20 patients from the ER every single day, no matter what.
Yeah, and you have to embrace that. And I think as a trainee, it is scary at first. Like, I will say, I thought I knew what busy was from the UCSF, from the San Francisco General County. And then I went to Bascom Palmer and their emergency department there. That's busy. Wow. That's busy. Having first year resident seeing 50 plus patients in a day. Right. That is like, I kind of had a new, you set your criteria for busy differently when you see that. But it's true. You have to be the type of person that embraces it and says, I want that. Like, I want to see more. I want to do more. I want to learn more. Yeah. It's hard to teach that.
It is. But I mean, it's the reason why it's the top program every year, right? It's a fantastic place. I'm glad you got to spend a year there.
It was a great year. I still am in touch with all the mentors there, and my co-fellows and I, we text on a daily basis. So it's a great network.
That's awesome. Well, Neel, I can't believe it. We've been chit-chatting for an hour, more than an hour.
Oh, wow.
We're having fun, see?
Yeah.
But I want to say I'm really impressed. I invite you on the podcast because I'm just impressed by all the work that you're doing. It's good to see these nice, new, young, energetic faculty members making big strides. I want to congratulate you. You're doing great work. Keep it up.
Thanks so much, Uday. And thanks for all the work that you've done. You're a staple of our daily routine for my wife and I, and for so many around the world. So thank you so much.
We all learn together, as you know. And I want to remind our listeners and viewers, remember, we got a new Cataract Coach podcast every week. Apple, Amazon, Spotify, Google, wherever you find your podcast, you find the Cataract Coach podcast. We also have the Retina Rounds podcast every Sunday, too. Fantastic. New Cataract Coach video every single day, and a new Retina Rounds video every single day. So until next time, catch you later.